Good morning, everyone. I would like to invite the next speaker of this session, Dr. Girija Goyal. Dr. Girija Goyal is an immunologist working with the organs on chip and diagnostic platforms to design better in vitro models of the human immune system and assays to test the performance of immunotherapeutics and vaccines. She is a senior scientist as a part of our advanced technology team in the Bio-Inspired Therapeutics and Diagnostics platform at the Weiss Institute for Biologically Inspired Engineering at the Harvard University. Her research focuses on the use of human lymph node organ chips to replicate and study human immune system's response to infection and vaccination. She is the recipient of Women in Immune Science Award 2022. Her talk today will focus on organ chips 2.0, hypoxia, microbiome, and immune cell integration in the human organ chips. Thank you.
Thanks to the organizers for this wonderful opportunity and to get to meet some colleagues here, as well as many wonderful students over the last few days. I'm a senior scientist at the Wyss Institute, the Hans-Georg Wyss Institute for Biologically Inspired Engineering at Harvard University. We like to have that mouthful there. I'm going to be presenting some work from my grants as well as John's grants here, but looking really at taking organ chips to the next level. Can we add immune cells? Can we add the microbiome? And to enable that microbiome integration, can we actually have oxygen gradient? So yeah, you've heard a lot about organ chips, microphysiological models, transfers, 3D cultures, and really the goal is to get clinical mimicry, right? So in our case, the examples that I'm going to show you, what we are trying to do is replicate patient genetics, largely by actually using patient-specific cells, add microbiome, add nutritional status, particularly for one of the stories that I'm going to be sharing, model the complex anatomical niche, be it vascularization, oxygen tension, mechanical forces, as well as the immune cells that are resident in the tissue. There is no tissue in the body, no site, which is devoid of immune cells, perhaps with the exception of the cornea. People say the brain or the testes may be immune privileged sites, but that's actually not true. You have different kinds of immune cells, some of which have adapted to be resident there. So the first story I'm going to show you is modeling the humoral immune response. Humoral in this context means secreted, and it's colloquially used for basically your antibody response, your B cell response. And 95% of the antibody-mediated protection that you're going to see in your lifetime is through T-dependent antibodies, T-dependent B cell activation, and that happens in an anatomical structure called the germinal center in the lymph node. This is where B cells, with the help of T cells, are going to edit their own DNA to make high affinity antibodies of different isotypes, as well as some of these B cells are going to go on and form plasma cells, which are long-lived cells that provide those antibodies over decades. In 80-year-old and above people, plasma cells producing antibodies against smallpox has been detected after 60 years. So they're still surviving for a long time. And this is really the holy grail of much of vaccinology, to produce these cells, which will provide you essentially with a lifelong immunity. We don't quite know yet why some vaccines do it and others don't. For instance, polio vaccine can provide this immunity for a long time, whereas influenza we take every year. Some of it is, of course, due to the yearly annual variation in the virus itself. But we also, including COVID, the vaccines actually wane over time, the protection they provide. Okay. So when we actually, when I set out to do this work as a postdoc, I had actually a more fundamental, basic question. I knew of groups who had been trying to recapitulate this in vitro, the germinal center function, starting from a tonsil or a lymph node. And my sort of question, which derived from trying to make a more useful product, was that I want to make this from blood, from circulating immune cells. So my sort of first basic question was that if I take immune cells from the blood that differ in composition, they differ in their differentiation status, can I actually mimic something of the tissue by actually changing their composition, so isolating different cells and putting them at a composition they would be in a germinal center, and by giving them a tissue-like density. So that's what I did here. I took T and B cells from peripheral blood, but I mixed them evenly, and I put them at a very high density in hydrogel. So this is like to the order of 200 million cells per ml. And then we seeded that gel here and then perfused the chip from the top. Where we had no idea what would happen, but as you see in the next slide, we serendipitously actually ended up nucleating germinal center-like structure. So I'm just going to take you through this slowly. So this is a chip where if I just seed it, but I never flow media through it, it just has media sitting on top of it, just like a static culture, and this is a chip under flow. And you can see these large aggregates of cells forming, which become AID positive. AID is activation-induced cytidine deaminase. This is an enzyme that B cells need to edit their own DNA, and it's only expressed in secondary lymphoid organs such as lymph nodes, spleens, etc. We also found that CXCL13 was upregulated, and this is a chemokine that's absolutely required to organize the germinal center in both mice and humans. And here we just quantified this over 6 different donors. You can see out of the 6, only 1 actually made these structures under static conditions, where under perfusion, all of them make these structures, and they are also significantly bigger. This is just showing you that the AID expression actually increases as the size of the lymphoid follicle increases. I've been speaking to a lot of students in the last few days, and we've talked about writing papers and review process, etc. So the reviewers did not like us calling it a germinal center because, of course, the lymph node has many, many different cell types. They were quite unhappy that we had the gall to call this germinal center or germinal center-like. So the paper is now published, and you will see we very carefully call it an ectopic lymphoid follicle. So in your immunology textbooks, a lymphoid follicle would be an emerging germinal center that is yet to be activated. So I told you earlier, these are also the structures where plasma cells get differentiated. So we wanted to test if that was going to happen. So to make things a little bit easier for ourselves, we used common laboratory stimuli, IL-4 and agonistic CD40 antibody, or heat-killed Staph aureus Coan1 strain. And what you can see is CD138 positive cells start emerging and are actually highly enriched in these germinal centers. So the first sort of bar we tried to cross with this to actually have a translational application is to look at influenza vaccines. Now, these are given seasonally in the United States and many other parts of the world, as well as many people are naturally infected every year. So this is what we would consider a recall antigen. It's unlikely that there's anybody in the population who hasn't seen some variant of influenza. So in this setting, you're really asking whether there are memory B cells present that can be activated. So we took 8 different donors, we seeded them on chip, and we compared the response against a human tonsil, which had taken from a child who had a lot of coughs and colds, and it was enriched in B cells producing anti-influenza hemagglutinin. This is a protein found in the influenza virus, IgG. So it's influenza-specific immunoglobulin that we are testing for. And all the 8 donors showed IgG production on chip, but you could see that some had almost 100-fold more IgG than others. So here I'm showing you one of these high responders and one of these low responders, respectively in the star and the triangle, and we tracked them over 12 days, and we found that wherever they start from, they continue to actually produce influenza specific IgG over time. We also checked whether the serum cytokines of vaccinated volunteers were recapitulated in this chip, and we found that as compared to some banked samples that we had from 7 different volunteers from David Walt's group, we made chips from 5 different donors, and all of them led to the upregulation of interferon gamma, IL-10, and IL-2 more or less consistently. We did see that the chips also increased GM-CSF, which we did not see in people's serum. So then we were charged by the Gates Foundation to actually see if we could reveal the efficacy of this qualine and water adjuvant, emulsion adjuvant. It's a low-cost adjuvant that's actually part of the Canadian COVID vaccine. But word of mouth was that they were unable to sort of replicate this in non-human primates. So here we took inactivated influenza, which is what most influenza vaccines are made of. We added either alone or with SWE, either in 2D or in chip. And as you can see, chips which are treated with SWE have the best antibody response. And this is coupled with an increase in these follicle-like structures here, although their size does not change. Additionally, CD138, number of CD138 positive cells also increases. So these are the plasma cells in the presence of SWE. And now we have been able to secure more funding from the Gates Foundation to continue this work with 6 different types of COVID vaccines. So this, up till here, we've published this. We now started looking at CD8 immunity, and this is just to show you in the first couple of donors that we can actually get CD8s that are specific to influenza. They are of different varying types, more central memory and effector memory, and we are so this is just more like proof of concept that we can actually find these guys. Most of our works, since we completed that earlier, and with GSK, and here I'm just listing the members of my team as well as GSK's team. So this is focused on different formulations of mRNA vaccines. So I just want to quickly walk you through what those formulations actually mean. So this is a conventional mRNA. It's that people first tried using for vaccines, particularly in the context of immunotherapy against cancers. The issue here is that your immune system has evolved to detect free mRNA and react to it in an inflammatory way and shut it down. So the problem with this is you get very low antigen expression. So one strategy people followed was to actually put an amplification sequence here that would lead to the RNA amplifying itself. So whatever few particles survive, they would amplify themselves and lead to better antigen expression. The other strategy that has been followed is actually to modify the bases to actually non-natural bases. This is the basis of the Moderna, Pfizer, most mRNA vaccines right now. They're the base modified. In both cases, this idea is to reduce the inflammatory response as well as allow the mRNA chance to translate. So to model this, we felt that we needed to model the vaccine site. And 95% of vaccines are given in the muscle. However, as many other speakers have pointed out, these are not high throughput systems. So they're very laborious. They take a lot of time. It takes us a whole incubator to do about 48 replicates. So we decided that it would not be a good strategy to actually couple this system to another chip So we actually built just a very simple 2D model of the human, of the muscle vaccination site, where we actually just co-cultured antigen-presenting cells such as monocytes and monocyte-derived dendritic cells in 2D with muscle cells and transfect them with the RNA vaccines one day before the APCs and the media is transferred over to the chip. So let me show you some data from this stage. So this is looking at a self-amplifying mRNA vaccine which encodes the rabies antigen as well as a GFP reporter. And as you can see, there is dose-dependent induction in muscle cells itself, right? So right now, I don't have any APCs here. And when I put APCs, in this case monocytes, with or without the muscles, what we see is that if the monocytes are alone, there are no GFP positive cells, suggesting that the APCs in the absence of muscle cells are unable to acquire the antigen, or either unable to acquire the mRNA or translate it. But in the presence of muscle, we can see that at least up to 100 nanogram per ml, there's a dose-dependent increase in the number of GFP positive monocytes, but then we start seeing toxicity after that, so the number crashes. So then we asked, what's going on here? So this is just the data I showed you. Depending on the donor of monocytes, we'll get anywhere from 10 to 20% of GFP positivity. We decided to do this experiment in a transwell, which had either 0.4 micron pores or 7 micron pores. What happens is when the muscle cells and the monocytes are separated by the 0.4 micron pore is that the cells can't come in contact. And what you see is you basically lose the GFP positivity. So we have not yet elucidated the full mechanism of what's going on, but we know that the APC and the muscle cells have to be in contact for the APCs to acquire antigen, suggesting that for this formulation, the APCs are unable to acquire and translate the mRNA themselves. We also see abundant cytokine production by all muscle cells and APCs alone, which further increases when the muscle cells and APCs are together. This is being depicted as a fold change in the presence of the mRNA, which is being delivered in a lipid nanoparticle as compared to the empty lipid nanoparticle. So next, coming to the chip, for which you earlier saw data with influenza, we again tested, we looked at the follicles, and across 4 different donors, we find again that the SAM-LNP increases the follicles, and we were also able to show an induction of anti-rabies IgG. This was a hallmark for us because in this case, it is actually a primary challenge. It's a naive antigen. Most people, most of our donors in the who are donating blood at the Brigham and Women's Hospital have never seen rabies. Okay, so then we additionally got funding from BARDA, but they had a very long-term goal in mind. They said, you need to make these chips last longer and have automated analysis of biomarkers because we want to send them to the moon or the International Space Station. So the stated goal of the project was they need to last 4 weeks and have automated biomarker detection. So then we modified this reservoir under which our chip sits such that instead of the I actually don't have a picture of the original reservoir, but you would have, these additional reservoirs where the media would come out. We removed those and actually put this microfluidic device on top of it such that each chamber fills every 2 hours. So this is a semi-discontinuous collection of media every 2 hours, which can then be transferred for any kind of analysis robotically. In this case, we actually coupled it to electrochemical sensors, which would then be exposed to this media in this automated fluidic handler. So with this challenge and with some media formulation changes, we were able to actually take our chips out for 28 days. And you can see right about the time when they start showing anti-rabies IgG is also when we see peak peak cytokine levels from many germinal centers supporting cytokines. Conversely, for cytokines that are antagonistic to germinal center formation, we start seeing a decline. Yeah, so in summary for this work, I've just shown you that here, trying to mimic actual lymphoid tissue, we have been able to show antigen-specific IgG against recall and naive antigens with 2 different types of vaccine platforms. I didn't show you all the data, but we've tested 5 different adjuvants, and we have optimized an intramuscular vaccination site model. So the reason I spent so long, one of the reasons I spent so long on that story is I want to stress, because I've had this question from students over the last few days, these are rationally designed, fit-for-purpose models where we are trying to change and tailor the cellular composition as well as the organs we are addressing based on the question. A sort of pipe dream of replicate, as in the case of the lymph node, we would not be able to have the 23 cell types in any way that would actually enable translational application. So you really have to choose your question as well as what parameters of that question you are considering. For instance, to model polio vaccine, instead of coupling the oral polio vaccine, instead of coupling the muscle, we actually couple the intestine chip to the lymph node chip. So it really depends, when we wanted to look at NK cells, we integrated those. We are also working at vascularizing this chip. Okay, so now I'm going to move on to a different chip where I will describe some of our microbiome and hypoxia work, and this is the intestine chip, with similar design, and you obviously heard this design being used in Dr. Ingber's talk, but in this case we are making use of vacuum channels placed to either side of the cell culture channels, which allow us to stretch the membrane on which the cells are being cultured. This is essential for forming these villi-like structures, and we have made this chip patient-specific by actually using patient-derived organoids, as I'll show you, which are then used to seed this chip. So before I get into that, I just want to briefly talk about organoids and organ-on chips. I believe this sort of message has been given by other speakers also. But there are pros and cons, right? So if I take organoids, they are very easy to handle. There are many companies that have now optimized 96-well plate analysis of organoids. However, the lumen, this is where your food, your microbes, your infections are going, is inaccessible, and most of these systems have proved resistant to vascularization. The chips are extremely expensive. They are very complex to culture. It's not, it requires a fair amount of training, even when postdocs join us, to get used to them. However, you can vascularize them, and the lumen, in the case of intestine, is completely accessible. So I'm going to first show you data from a Gates Foundation project in collaboration with the Aga Khan University in Pakistan, looking at environmental enteric dysfunction. This is a disease which is largely seen in areas where poverty, malnutrition, low access to water, clean water, and medication are found, and it leads to stunting of the children, not surprisingly, cognitive issues, and many other issues such as low efficacy of vaccines. So to mimic this, what we wanted to do, we first took just healthy control organoids. These we were able to secure from the Children's Hospital, and then we took EED organoids from a group in Aga Khan. So these are actually biopsies from children with EED, intestinal biopsies, and then they are allowed to culture in organoids, which can then be maintained. And then to mimic malnutrition, we actually gave them like complete media, or we gave them media which was depleted in niacinamide and tryptophan. And as you can see here, when I give them media depleted in niacin and tryptophan, they don't actually die. There is a layer of stem cells which is still maintaining mostly intact barrier left behind. And you see this both for the healthy chip and EED chip. However, when you look at the transcriptomics, you see that if you compare to a clinical signature of EED, so this is just transcriptomics from patient biopsies, and you look at trying to replicate it, if you just have the patient cells but in complete media, you don't replicate it. If you have nutritional deficiencies, even in healthy controls, you don't replicate it. However, you start seeing replication when you have both nutritional deficiencies as well as the patient background. So next but this is a complex syndrome. Immune cells are involved, microbiome is involved, so we wanted to be able to culture both diseased and healthy complex microbiome from human stool in these chips. To do that, we had to mimic the oxygen gradient that's seen in your intestine. In the lumen, the oxygen tension is very low, ranging from microaerophilic 5% to completely anaerobic, whereas of course on your body side, the normoxic tension of 21% is maintained. So to do that, we actually build this chamber where the upper channel and bottom channel of the chip are fed by completely different air streams as well as media streams, and you can see just the different channels and the different colored media. And we looked at what flow rate of nitrogen being flowed in the apical chamber will actually give us what kind of oxygen tension. So you can see that. So if I push nitrogen apically, I can drop the oxygen tension down. We measured this by implanting these oxygen sensitive sensors onto the chip. And what you can see is, unlike aerobic conditions, which is here, once I start this anaerobic culture, both all in the inlet, middle, and the outlet. So inlet being where the media is entering, middle of the chip is being here, and outlet, I can achieve quite low oxygen tensions, even lower than 1%. So using this, we tried to take human stool and culture this on this chip, and we found that we were able to maintain a fairly diverse set of bugs. So we recovered about 200 unique OTUs from this chip. So coming back to environmental enteric dysfunction, so we now wanted to ask what the contribution of the dysbiotic microbiome from EED patients will be. So I'm now looking at this, it's just called the Gordon microbiome after the researcher who defined this consortium. I'm looking at what happens to the cytokine levels when I expose healthy chips or two EED donor-derived chips to the microbiome. And as you can see, the EED-derived donor chips are much, much more sensitive to that microbiome than the healthy chip, even though this is a disease microbiome. Similarly, for nutritional deficiency, the EED genetics are playing a role because you can see these donors are way more sensitive. And when you actually combine both insults, that is nutritional deficiencies, as well as the disease microbiome, the chips, the EED-derived chips just go completely crazy with the high level of inflammation. Not surprisingly, this high level of inflammation leads to the barrier breaking. We are measuring apparent permeability here. This does not happen in healthy chips, as I just showed you with the diseased microbiome. It also does not happen in healthy chips with the healthy microbiome. So we just have duodenum or ileum chips where we just use a normal infant, this is called the baby biome, as well as a probiotic consortium called VSL3. As expected, because the disease microbiome does not inflame healthy intestine chips, it also does not cause any barrier break. We've also tried to integrate immune cells here. Some of our work with PBMCs is published. I just wanted to show you some DC integration. This is just a villus that has toppled over in the course of imaging. And I'm just going to, in the interest of time, not run through the whole, well, I guess I have to run through the whole thing. So you're going bottom up through the membrane into the villi, and you can see dendritic cells that I seeded at the bottom have now gone all the way up into the villi. So with this, we have started doing polio vaccine studies, and where we have been able to show that when you infect the intestine chips with Sabin 1, or I forget, the Sabin 1 or 2 strain, you can see that the strain replicates both in enterocytes as well as in dendritic cells, and leads to production of further live virus. We have a similar project like that looking at bacterial vaginasis in the female vagina and cervix chips, where we are trying to look at what the effect of the vaginosis is on fertility, on sexually acquired infections, and many other diseases. And this work, again, is funded by the Gates Foundation, and this is the team that is involved. What is very exciting about this project is that we have come close to sort of our ideal goal, that these chips are being conducted in parallel to the clinical trials. So in fact, data from these chips will actually tell us which formulation should be taken to the clinical trials, because they replicate both the healthy and the diseased microbiome quite well, and the phenotypes of bacterial vaginosis. So yeah, here I will end. My email ID is here. I'm also very active on LinkedIn, so you can find me. And I would just also like to add that we are recruiting postdocs with gene editing, immunology, or microbiology experience.
Thank you, Dr. Girija, for the very insightful talk. Now we can take a few questions.
And images, and I like the humility with which you said that you will not be able to build the entire lymph node. I like it. I'm an immunologist myself. So one question on your vaccine challenge studies in the lymph node. You were actually giving the mRNA vaccines. Did you have a chance to test any vaccines that are vector mediated, like adenoviral constructs or anything? And number one. Number two is, in your vaccine challenge studies, did you also have dendritic cells and other antigen presenting cells present these things?
Yeah.
Number two. Thank you.
Yeah. So I'll answer, my apologies, I always forget this in my talks because of course I'm so familiar with the work. So to do all the vaccine related work, we did have antigen presenting cells embedded in the germinal centers. Yeah, what was your first question again? Can you repeat?
No, did you use the vectors or vaccine constructs to actually challenge your germinals or other follicles? Not just the mRNA vaccines. Did you use any other vaccine?
Oh, I see. No, so currently we've just tried either recombinant protein or inactivated virus in the case of influenza. We also have some sort of cutting edge DNA origami type of vaccines that we are trying. And then we have tried the mRNA. We hopefully will be able to secure some adenoviral COVID vaccines for our upcoming grant. Or if you have access to them, I would love to collaborate. But we do need, we currently don't have access to any vector-based vaccines. Yeah.
So also being a veterinarian, I have a very strange question for you. There is a current outbreak called lumpy skin disease that's happening throughout the world. A very strange disease, pox viral type. So is there any model that you're thinking? I'm thinking about a model where there is a transwell with keratinocytes, melanocytes, skin cells, and then the lymphatic cells that can be on the well, and you collect the supernatant. That's one model. But can you think about a chip that can actually solve this problem of understanding how the pox, you had done something with pox, right? So can you think about something that can, because this is a very acute outbreak there.
Yeah, so I think it really depends on your question. So if you're interested in viral infection and evolution, I think the skin, as you described, would be a great model. If you're interested in whether antibody or T-cell response is being formed, you do need a secondary lymphoid tissue type of condition. It's very hard to get both T and B cell responses against recall antigens in 2D. So that's data that has been published. One of the things we have never attempted to do, but just to continue that thought, is that now even if you have the T cells and the B cells forming, how do you know they're going to be effective or what they're going to do? You need to now take them back to the site of infection, which is something we hope to do if we ever get funded for that, yeah. So it really depends on your question, I would say.
Thanks, Girija. I have more questions, but I'll connect on LinkedIn. Thank you.
Excited. It was a fantastic talk and some fantastic results to look at and giving us the confidence that we are going to see these things soon, with pharma companies evaluating it for clinical trials. So my question to you is that definitely your EED data that you showed, where you put all the players together, the microbiome as well as the immune cells, and then you have measured the cytokine profiles, and that shows you that it is very, very close to what you observe clinically. So my question to you is regarding the bacterial vaginosis thing, because I come from a reproductive health institute and I'm an immunologist by training. So I feel that is going to be a wonderful thing. My question to you was, are you going to have the autologous microbiome of the women also from the clinical trial along with their vaginal samples?
Yeah, so we've, in that study, we are actually just using commercially derived or just random hysterectomy samples that we've been able to acquire to get the cells, at least for the cervix. Of course, you can get the vagina from the hysterectomy. Our hope is with African collaborators to actually be able to get some cells. However, if you get a swab or even a biopsy, expanding, it's a lot of work. Even for organoid type of work, we actually depend on our collaborators who are more experienced in actually passaging these cells and getting the right things to grow out over years time frame. So we've not really focused on, in that setting, we have cells from different ethnicities, but they are not from the trial patients. But we are getting the microbiome from patients. So that we've been able to already start that work from local sites, local to us, MGH, where we're able to get the bacterial vaginosis samples, and now hopefully we'll be able to get African samples in the next year or two.
Can I just have a quick question?
Sure.
So when you are doing these studies, say for 48 hours in EED, you showed that it went up to 28 days, right?
Yeah.
So when you are adding the microbiome, have you checked the composition? Does it change?
Yeah. So these are in vitro, just to remind everybody in the audience, this is an in vitro system, and of course what grows is dependent on what you provide it. So this is why the hypoxia was so important. So for most of our studies, we'll actually only push the microbiome to 3 to 5 days, because after that it just eventually you overproliferate. And what we do find is depending on the bacterial consortium you have, if you have E. coli and enterococci who are sort of more permissive, will grow everywhere, they tend to start overtaking the culture right about that time. This obviously has not stopped us from revealing clinically relevant phenotypes, but it is something that we are now sort of, I think we had this discussion earlier, we are figuring out how much resources to devote to actually try to maintain not just diversity in terms of species, but proportions over time. But yes, intestine, for sure, enterococci will grow out. In the vagina and cervix, lactobacilli are very stable, as are Gardnerella and things like that. So there we've had more success replicating for a longer time, but that's because we don't have E. coli in that mix. I'm pretty sure if the day we added E. coli, they would just take over.
Hi Girija, I have a question here. Very interesting talk. I just wanted to know whether you have done any drug testing in the organoid versus organ in a chip, because many of the private companies are coming up with drug testing in spheroids or organoids. taking into consideration the complexity itself is different when you make an organ in a chip. So won't you think that that would be better than the spheroid testing?
Yeah, it's sort of a self-fulfilling prophecy in our case because we are using organoids. We can check for barriers. As I showed you, we can do microbiome culture. So the people who are the companies we have ended up working with, many of them are typically they're looking for drugs against the microbiome. So that's the people who are going to come to us. If they have separate pipelines, highly established pipelines of organoid work, etc., where they're going to take 10,000 hits and get it down to 10 or 20, and then the top 4 or 5 will end up coming to us, keeping in mind we are a non-profit institution and we want our postdocs to get first author papers, and also we are not like a service provider, but that's the pipeline that's emerged. You cannot eliminate, and you shouldn't, the high throughput system, but once they have the hits they want to take into clinical trial, that's when such a complex model, but it's going to, each chip is like a mouse, right? It's going to give you lots, it's going to tell you if it saved you from barrier, was Zo1 disrupted, which microbial species grew out in the presence of the drug. So yeah, so that's the kind of testing we are doing of basically your prioritized hits. We are not, and most companies who work with this kind of chips are not at the scale where you could do 1,000 drug library. Yeah.
Right. So we'll just have one quick, if you could just ask quickly once, and then later on if there are any questions. Actually, we are still kind of running out of time, so maybe just one question here, and maybe you could catch Dr. Girija after the talk.
Ask about the lymph node on the chip model. So the lymph node on the chip model is not having, as you said, all the cells you cannot incorporate into a single model, but along with that it's lacking some essential structural components like high endothelial venules, etc. So how are you addressing that problem?
Yeah, like all of research, we are under constant pressure to publish and have our people well situated in their careers and all. So just like all of you, we are making practical decisions, right? So for us, vascularization is easiest and very impactful. So we are looking at knocking out the HLA locus so that we can basically have a universal endothelial cell that we can use. We are constantly asked about follicular dendritic cells or reticular cells, but to get this is immune chip, so it has to be autologous. To get those in an autologous way would be very difficult. So it's really, that's why I stress the fit for purpose. This is a huge advance on being able to test vaccines preclinically. And we don't need those cells for that, but when we start talking about trafficking, we do need an endothelial barrier. So for us, that's the highest priority, to get that endothelial barrier in there.
Sure.
Thank you.
Thank you, Dr. Girija, for the very interesting talk, and I'm sure there are more questions, but maybe you can catch her after the talk and then ask her. So yeah, thank you.